So, polygenic inheritance. Mendel's studies mainly describe those traits that have distinct alternative forms, such as flower color, which are either purple or white. But if you look around, you will find that there are many traits which are not so distinct in their occurrence and are spread across a gradient. For example, in humans, we don't just have tall or short people as two distinct alternatives, but a whole range of possible heights. Such traits are generally controlled by three or more genes and are thus called as polygenic inheritance. Polygenic traits. Besides the involvement of multiple genes, polygenic inheritance also takes into account the influence of environment. For example, human skin color is another classic example for this. In a polygenic trait, the phenotype reflects the contribution of each allele. That is, the effect of each allele is additive. To understand this better, let us assume that three genes A, B, C control the skin color in humans, with the dominant forms A, B, and C responsible for dark skin color and the recessive forms ABC for light skin color. The genotype with all the dominant alleles AA, BB, CC will have the darkest skin color and that with all the recessive alleles AABBCC will have the lightest skin color. As expected, the genotype with three dominant alleles and three recessive alleles will have an intermediate skin color. In this manner, the number of each type of alleles in a genotype would determine the darkness or lightness of the skin in an individual. Now, pleiotropy. We have so far seen the effect of a gene on a single phenotype or trait. There are, however, instances where a single gene can exhibit multiple phenotypic expressions. Such a gene is called a pleiotropic gene. The underlying mechanism of pleiotropy is, in most of the cases, is the effect of a gene on metabolic pathways which contribute towards different phenotypes. An example of this is the disease P. phenylketonuria, which occurs in humans. The disease is caused by mutation in the gene that codes for the enzyme phenylalanine hydroxylase. Single gene mutation होता है इसमें. This manifests itself through phenotypic expression characterized by mental retardation and a reduction in the hair and skin pigmentation. So this involved error of metabolism is also inherited as the autosomal recessive trait. The affected individual lacks an enzyme that converts the amino acid phenylalanine into tyrosine. As a result of this, phenylalanine is accumulated and converted into phenyl pyruvic acid and other derivatives. Accumulation of these in the brain results in mental retardation. These are also excreted through urine because of its poor absorption by the kidney. Sex determination. The mechanism of sex determination has always been a puzzle before the geneticists. The initial clue about the genetic or chromosomal mechanism of sex determination can be traced back to some of the experiments carried out in insects. In fact, the cytological observations made in a number of insects led to the development of the concept of genetic or chromosomal basis of sex determination. Henking 1891 में was able to trace a specific nuclear structure all through spermatogenesis in a few insects, and it was also observed by him that 50% of the sperm received this structure after spermatogenesis, whereas the other 50% of sperm did not receive it. So Henking gave a name to this structure as the X body, but he could not explain its significance. Further investigations by other scientists led to the conclusion that the X body of Hanking was in fact a chromosome, and so it was given the name X chromosome. It was also observed that in a large number of insects, the mechanism of sex determination is of the XO type, that is, all X pure and additional X chromosome besides the other chromosomes, that is, autosomes. On the other hand, some of the sperms bear the X chromosome, whereas some do not. Eggs fertilized by sperms having an X chromosome become females, and those fertilized by sperms that do not have an X chromosome become males. Do you think the number of chromosomes in the male and female are equal? Due to involvement of the X chromosome in the determination of sex, it was designated to be the sex chromosome, and the rest of the chromosomes were named as autosomes. So grasshopper is an example of exotype of sex determination in which the males have only one X chromosome besides the autosomes, whereas females have a pair of X chromosomes. So these observations led to the investigation of a number of species to understand the mechanism of sex determination in a number of other insects and mammals, including man. XY type of sex determination is seen where both male and females have same number of chromosomes. Among the males, an X chromosome is present, but its counterpart is distinctly smaller and called the Y chromosome. Females, however, have a pair of X chromosome. Both males and females bear same number of autosomes, hence the males have autosomes plus XY, while females have autosomes plus XX. In human beings and in Drosophila, the males have one X and one Y chromosome, whereas the females have a pair of XX chromosomes. A pair of X chromosomes means XX chromosomes, besides the autosomes. So in the above description, you have studied about two types of sex determination mechanism, that is exotype and XY type. But in both the cases, males produce two different types of gametes. First is either with or without X chromosome, or some gametes with X chromosome and some with Y chromosome. Such type of sex determination mechanism is designated to be the example of male heterogamety, because in this male is deciding the sex of the progeny. In some other organisms, just like birds, a different mechanism of sex determination is observed. In this case, the total number of chromosomes is same in both males and females, but the two different types of gametes in terms of the sex chromosomes are produced by females, that is female heterogamety. In order to have a distinction with the mechanism of sex determination described earlier, the two different sex chromosomes of a female bird has been designated to be the Z and W chromosomes. In these organisms, the females have one Z and one W chromosome, whereas the males have a pair of Z chromosomes beside the autosomes. So now sex determination in humans. It has already been mentioned that the sex determining mechanism in case of humans is XY type. Out of 23 pairs of chromosomes present, 22 pairs are exactly same in both the males and females. So these are the autosomes. A pair of X chromosomes are present in the female, whereas the presence of an X and Y chromosomes are determinant of the male characteristic. During spermatogenesis among males, two types of gametes are produced. 50% of the total sperm produced carry the X chromosome, and the rest 50% of the has the Y chromosome besides the autosomes. Females, however, produce only one type of ovum with an X chromosome. There is an equal probability of fertilization of ovum with the sperm carrying either X or Y chromosome. In case the ovum fertilizes with the sperm carrying X chromosome, the zygote develops into a female XX, and the fertilization of ovum with Y chromosome carrying sperm results into a male offspring with XY chromosome. Thus, it is evident that it is the genetic makeup of the sperm that determines the sex of the child. It is also evident that in each pregnancy there is always 50% probability of either a male or a female child. It is unfortunate that in our society women are blamed for giving birth to female child and have been ostracized and ill-treated because of this false notion. So sex determination in honeybee. Sex determination in honeybee is based on the number of sets of chromosomes an individual receives. An offspring formed from the union of a sperm and an egg develops as a female, queen or worker, and an unfertilized egg develops as a male drone by means of parthenogenesis. This means that the males have half the number of chromosomes than that of a female. The females are diploid having 32 chromosomes and males are haploid, that is having only 16 chromosomes. This is called as haplodiploid sex determination system and has special characteristic features such as the male produces sperms by mitosis. They do not have father and thus cannot have sons, but have a grandfather and can have grandsons. However, how is the sex determination mechanism different in the birds? Is the sperm or the egg responsible for the sex of the chicks?